Smoking is a major public health problem worldwide. There are approximately 50 million people in America who are addicted to some type of tobacco product, including cigarettes, cigars, chewing tobacco, and snuff. It is of utmost importance that the public understands the risk behind developing a nicotine addiction, yet it cannot always be controlled. Some biological factors that influence nicotine dependence are genetics, age, and susceptibility to depression. Biological factors have the capability to enhance one's chances of becoming reliant on nicotine. Though nicotine addiction is extremely prevalent across the globe, less than 10 genes with genetic variations are directly related to addiction. In a study conducted by Rebecca Perez Morales, a chemical scientist at the University of Juarez, her results concluded that it is possible that carriers of the ChRNA3 SNP are more susceptible to diseases related to tobacco consumption through nicotine dependence. In genetics, a single nucleotide polymorphism is a substitution of a single nucleotide at a specific position in the genome. The ChRNA3 gene, rs1051730T allele, was very significantly associated with increased risk of nicotine dependence. The ChRNA3 gene provides instructions for making one part of a larger protein called a neuronal nicotinic acetylcholine receptor, NACHR for short. In the brain, NACHR proteins are widely distributed and play an important role in chemical signaling between nerve cells. The channels also open in response to nicotine, the addictive substance in tobacco. That is why it is essential to test the ChRNA3 gene and examine its effects. In order to test your chance of developing a nicotine addiction, you must take a SNP chip test. Our test will include all 8 genetic variations. SNP rs1051730 is a SNP in the nicotinic acetylcholine receptor alpha 3 subunit of the ChRNA3 gene. The T allele is very significantly associated with increased risk of becoming reliant on nicotine. If someone has this allele, they are not at higher risk for becoming smokers, but if they do smoke, they are at higher risk for becoming dependent on it later and developing lung cancer. The CC genotype is associated with a 0.8A increased risk of disease and cancer. It does make sense that genetic variations in this gene impact risk for nicotine dependence because the T allele reports increased risk of nicotine dependence. Genetic testing is a helpful way to identify genetic variations in a specific trait that are linked to diseases and disorders. This technology is beneficial because it can aid in preventing harmful diseases and disorders, knowing if the gene will be passed on in future generations, and emotional relief of knowing the intensity of the variation. Prevention can include uptake in physical activity and smoking discontinuance. In addition, for some heritable conditions, genetic testing can provide powerfully predictive information about the individual's future health status. Limitations include having an increased risk for nicotine dependence if you test for one or more variants. An increased risk is not a guarantee you will develop this condition. Other factors such as substance use, age, depression, or other mental illnesses may increase your risk. Test errors may occur. In this case, we will notify and retest you. After having a positive genetic test for a gene, the likelihood of developing the disease in this space does increase. However, the amount it increases varies depending on the different variations of the genotype. For example, a person could have one gene for a normal cell and one for the disease, which would cause a smaller increase in the likelihood than if the person had 2 recessive disease cells. These different versions will result in a different percent chance that the person will develop this trait. This can be shown on SNPpedia. For example, with the rs1051730 gene, when having the mixed genotype resulted in a 1.3% increase, while having 2 copies of the diseased allele resulted in a 1.8% increase in developing the trait. Genetic tests can cost anywhere from $200 to over $2,000, which can create significant financial hardships and stress. If a person were to pay for a test and a variation was identified, they could be left with no financial backing to cover treatment costs. Accuracy is also not fully guaranteed. In addition, it is concerned that mandatory testing can violate individual privacy rights and lead to discrimination in parts of their lives, such as in the workplace. Genetic and genomic information is complex, and health risks associated with its information are often